All right, I think we can start with our first speaker of the morning session on ARDD5, Anja Groth, here from the University of Copenhagen. Whenever you're ready, Anja, go ahead.
I'm ready, yeah. Thank you, Morten, and thanks to all the organizers. It's really a great pleasure to be here. It's been a fantastic meeting, and I learned a lot over the last couple of days. And I really appreciate to get this chance to talk about our work, and this is going to be kind of a deep dive into the mechanisms that maintain epigenome fidelity in our cells, and therefore also maintain cells fit over lifetime. So in my lab, we're interested in understanding how epigenetic information is propagated during mitotic cell division, and trying to understand how, in this relation, cell proliferation affects cellular plasticity and also epigenome fidelity. So basically, epigenetic cell memory underpins human physiology and health very broadly. And obviously, every time a cell divides, it takes a decision of duplicating its parental ID or undergoing a programmed change in ID. And when these processes fail, you can have unscheduled changes that leads to adverse loss or gain of cell functionality, which is what we see in cancer and aging. And of course, as we also talked about a lot about at this meeting, you can also bring back cells from either a differentiated state or from an aged state to a younger or more pluripotent state. And again, in all these decisions, establishment and maintenance of chromatin states are really important. So, however, while we understand very well how DNA is replicated, how mutations in DNA are repaired, we still don't understand relatively little about how the epigenome or chromatin states are actually inherited to daughter cells. And I think this is really a fundamental knowledge gap, which is limiting our understanding of how epigenome evolves in development, cancer, and aging. I mean, given the huge number of cell division that takes place across human development, both during early embryogenesis, but also later in life to counteract aging. And so chromatin organization is a complicated structure, and we are in particular in love with the nucleosome, which is the basic unit of chromatin, where you, in addition to DNA methylation, you also have the histones that can carry a large variety of post-translational modifications. So these histones and DNA modification landscapes are highly cell type specific, and we know that these chromatin structures and these modifications are deteriorating with age. And so what we are trying to understand is what happens to these, the epigenome every time a cell divides. So basically, every time a cell goes into S phase and has to replicate its genome, replicate DNA, this leads to a genome-wide disruption of the epigenome. So basically, ahead of the replication fork, the basic structure of chromatin is disrupted, and the major challenge for the cell is to restore this environment and transmit it to the daughter cells in order to maintain epigenome fidelity. And this is what we're trying to understand. And so, in order to do so, we developed a number of new technologies to study how chromatin is replicated and how chromatin patterns and structures are inherited to daughter cells. So for this, we use incorporation of nucleotide analog. It's like a pulse. And you can then isolate this replicated DNA and follow how histones, marks, or DNA modifications are inherited across cell division. And so this means we can basically track both histone and DNA modifications, and also chromatin accessibility, etc., and the inheritance to daughter cells. And so what we've learned from these studies about the inheritance of histone modifications, I'll try to just briefly summarize here. So basically, when the replication machinery hits or moves in chromatin, all the nucleosome structures will be disrupted, and there is a process that allows these histones with their modifications to be recycled onto the two daughter strands. And they're recycled they maintain all their modifications, they maintain their position in this process, And they also end up being distributed relatively equally to the two daughter strands so that this information can be inherited to daughter cells. So this is all, you could say, a mechanism that really preserves the original environment. But then you have this highly disruptive event, which is the incorporation of new histones. And this is, as I said, highly disrupted. It dilutes the whole epigenome by twofold. And the challenge for the cell is now to modify these new histones so they look like the old ones in the area where they were incorporated. And this is a process which is uncoupled from DNA replication, and it's actually a huge challenge for the cell, and as I will show you, this takes place over almost the full cell cycle. And it's also a very heterogeneous process. So this is what we now know about how histone modifications are inherited. So if we look at the DNA modifications, I mean, I'm sure you're all aware that at the time when DNA is replicated, you have the symmetric methylcytosine is now distributed on the two daughter strands with the parental DNA. And the cell now has a huge machinery that can now copy, in a copy-paste mechanism, this information to the newly synthesized strand. What we've been interested in, and this is work by a fantastic postdoc who now runs her own lab at Copenhagen University, together with Petra Haikova's lab, and we've been interested in understanding the kinetics of this. How long time does it take for the cells to restore DNA methylation patterns after replication? And what's the fidelity in the process? And so what we could see is that after replication for a period of time, the DNA stays hemimethylated. So this copy process is not an instant thing. It takes time. Actually, it takes several hours, up to 4 or 5 hours, and before you have restored the full pattern of DNA methylation. So this means it continues into the next cell cycle and in daughter cells. And in addition, we see that while most of the new methylation is added to the newly synthesized DNA strand, there is also addition of marks to the old strand, which tells you something about the fidelity in this process. This is not a direct copy-paste thing. There's also new methylation coming on, probably because it's never fully finished just within one cell cycle. And the same thing actually goes for hydroxymethylation, which is also very slow restoring, but doesn't have a proper copy-paste mechanism. So the reason why this is important is really to understand that every time a cell goes through the process of replication, which means in every cell division, you have a dilution of all the histone modifications, and this takes a long time for the landscape to restore. Some marks associated with transcription are actually very fast, but the marks that we are often interested in terms of epigenetics, and also which we've seen a lot of talks about how they deteriorate, these repressive marks, they're very slow and takes a full cell cycle. They're almost only restored before the next round of replication. And the same thing is true when we look at the DNA modifications. So this illustrates a bit how huge a challenge cell proliferation is to the epigenome. And it seems that the system has kind of chosen to value plasticity, which is of course what is introduced by diluting out your whole epigenome, at the expense of fidelity. And when you think about how long time and how much effort the cell has to put into restoring the epigenome, I think you can easily imagine that if under this process the cells are challenged with stresses or metabolic changes or limitations, you can easily imagine how this process may actually not be completed, and that would contribute to deterioration of the epigenome. And so now I'm going to shift gears a little bit and talk about some more recently published work, and which really deals with the question of whether is it actually important to inherit histone modifications. I mean, we've also seen work here where you can express the Yamanaka factors and you can basically rejuvenate the cells. So maybe everything is already known and you actually don't need to inherit these. And the reason why this is something that we haven't been able to address for a very, very long time, but with recent advances from my lab and the lab of Stingoy Sang, which has basically identified loads of components in the machinery that replicates DNA, which are also responsible for transmitting the parental histone. So basically, the cells have chosen to couple epigenetic and genetic inheritance in the same machinery. And so, and actually there are two different, there's a mechanism for replicating the leading and the lagging strand. There's also two parallel pathways for segregating histone-based information. And this means that we can now go in and disrupt one of these pathways, and we can start asking what happens to cell if chromatin replication, or if cells can actually not inherit its epigenome correctly. And what we did is we made a mutation which allows us to force all the old histones to one strand and make the new strands composed mainly of newly synthesized unmodified histones. And we've been asking, does that actually affect cell identity and functionality in mouse CS cells? And this has been a great collaboration driven by Alice, Alva, Nicolas in my lab, together with Alba in the lab of Josh Strickman, and also in collaboration with Robin Anderson. And what we wanted to ask was, of course, in these mutants, what happens to the epigenome? So we looked both in this histone recycling mutant, and then we took this mutant cell line as well and generated cells where we had basically reverted these mutations back to wild type, so rescue lines, trying to address, so if you first derail the epigenome, are the cells actually able to restore them again? And in that case, how? And so when we look at what happens in these cells, we can see that the fidelity of the epigenome is challenged very dramatically. So we see increase in K27 trimethylation. This comes not at the sites where the cells are supposed to have this marked. No, this comes at aberrant loci across the genome. So the cells basically provide a blanket of these modifications at sites where they shouldn't be. And as you can see here, it's fully rescued in this rescue line. And there's also, this is a complicated plot, but what it basically shows is also at all the sites where you're supposed to have these modifications, you now get a total shift. So you basically, you're not able to uphold the proper landscape also at promoters, and this leads to changes in gene expression in these cells. So that was K27. What happens now to K9? And we basically see the same thing. Again, we see an increase in the background level of K9 trimethylation. And this basically comes because these cells are losing, so these are reads that are mapping to repeats. And you can see that in the mutant, you're basically losing this modification from repeats. And this is fully restored in the rescue line. So this means that the cells are no longer able to, and maybe I should show this slide here. So you can see that K9 trimethylation is lost broadly across repeats in these cells. And you have deregulation or desilencing of a large number of repeats, mainly the LTRs. And all this is fully rescued in the rescue line. So this is something that gets to be an aberration, but as soon as you turn back proper inheritance, you're now maintaining these patterns. And basically what this is showing is, again, it's supporting the idea of having this blueprint where you copy marks from new to old histones, and it has to happen on both strands. But I think what it also shows, which is, I think, very new understanding of how epigenetic noise can arise. Because what we see is that the proper handling of histones at replication forks is contributing to reducing epigenetic noise. So once you start messing up these inheritance systems, the cells start accumulate epigenetic modifications in the wrong places. And so this argues that if you have defects in chromatin replication, it can broadly derail the epigenome. And of course, what we're interested in understanding is whether this is a cause of epigenetic decline in cell aging. And we believe that these histone recycling models present a super attractive model system where we can now start to study epigenetic noise and how it's linked to cell proliferation, and also start asking questions about how does epigenetic noise actually impact on cell functionality and fitness. And if we look at these mouse ES cells and ask whether they maintain their identity under these conditions, we can do that in chimera assays where we are asking about their pluripotency and their ability to contribute to the early embryo. And what we can see here is that the mutant cells are unable to contribute, but the rescue line fully restores this function. So as soon as you get back proper inheritance, you also restore the epigenome gene expression and functionality of these cells. And I think, yeah, so this argues, of course, that correct inheritance of modification in each and every cycle is important, but it also tells you about something about the plasticity of the system, which I think is consistent with what we're seeing in the reprogramming experiments, where cells are actually basically able to correct their epigenome and where this information is coming from and how it is stored. I mean, I'm sure it's partly as in the genome, but also probably in other epigenetic mechanisms. And so I'm just going to mention one last thing at the end, because I talked a lot about how this works if you make a mutant. And so obviously cancer cells or aging cells don't necessarily make this type of mutant in the histone recycling machinery. But what we also know, and this is work that we've done together with Nitika Tanaya, it was driven from her lab, and she's at the Erasmus MC in the Netherlands. And this was trying to understand, so what happens in the situation where cells meet replication stress? And as you know, if cells are exposed to various types of stresses, it turns on new sets of genes, and a lot of transcription happens, and this leads to replication-transcription conflicts, which leads to arrest and damage at the replication forks. And what we've been able to show is that every time a fork encounters problems, you basically accumulate K9 trimethylation at these forks. So this is a protective mechanism. It ensures recruitment of repair proteins and fork restart. And most of the time, as soon as the forks start again, you totally remove this. So it's a transient thing. But obviously, over time, patterns like this could be stabilized. Plus, it also diverts the attention of these enzymes from their proper task, which is to maintain the epigenome as the cells divide. And so we think that replication stress is likely a source of epigenetic infidelity in both cancer and aging. And these are some of the directions that we are now trying to explore. And with that, I'm going to finish. I think I thanked my lab and my collaborators and all our funding sources. Thank you.
Thank you so much, Anja. We have time for a few questions. Maybe. Okay.
That is very nice. I was wondering if there is this infidelity and you get these aberrations, then in fact that should be a single cell phenomenon. No? I mean, they must be different from cell to cell.
Yeah.
And also that information must be permanently lost because there's no memory of it, correct?
Yeah.
If you have...
It's not lost. So this is clearly what we say. It was very surprising. So we are not generating new epi-alleles in that way. So things that get stabilized in new epigenetic states. And I mean, what we think is going on is that so in epigenetics, people have tended to separate establishment from maintenance. But for many parts, the establishment signals, so signals to induce silencing at repeats, for instance, is there all the time, for instance, through some of zinc finger proteins and other things. It's just not alone enough. You need the recycling and the memory as well. So I think that's partly what is explaining why you can get the things back in the right place. And we did do single-cell RNA-seq. I didn't go through it here. And what we can see is that the cells, especially in S phase, they start expressing genes that they shouldn't do. And yeah, so there's more heterogeneity when they are in this pluripotent S phase state. Yeah, that's true.
Thank you. What an amazing talk. Thank you. I just wondered, would you expect to see different histone noise in different tissues that proliferate, like, say, the gut, and proliferate all the time?
I think this is a great question. So this is obviously done in mouse CS cells, and we are now aiming to make models where we can actually look at different cell types and see how, yeah. So yes, I think it would affect the epigenome differently, and the genes that will be affected will be different, and also the types of repeats. Yeah, so I think so.
All right, thank you so much, Anja. That was a really fascinating talk.
Thank you.